Hello, I'm delighted to share with you today the journey that we've been on here in Cork, in the very south of Ireland for well over a decade now, implicating what's going on in our gut with what's going on in our brain. And here are my disclosures. Nothing significant related to this actual presentation. So this is a story I like to tell. And this is the story of this painting called Le Bateau. And it's by Matisse. And it hung in the Museum of Modern Art as part of a retrospective exhibition in 1961. And it hung like this. And it wasn't until a very eagle-eyed stockbroker called Geneviève Habert noticed that something was wrong with the painting. And what was wrong with it was that it was hanging upside down. And so sometimes you have to turn things around for you to be able to see them in the right way. And as a neuroscientist, that's kind of what my take-home message for this talk is, that sometimes we have to look at things through the gut. And of course, this goes back to Hippocrates, who's thought to have said that let all diseases begin in the gut. Now, that can be quite surprising when I talk to my psychiatry and neurology colleagues. And all we have to do then is look to our language. We use phrases like gut feelings, gut instincts. We make gutsy moves when we're brave. We're gutted when we're disappointed. We have butterflies in our tummy when we're nervous. So we personify our emotions in the gut. And in terms of aging and the gut, this is one of the key people that many of you will know, and that is Eli Metchnikoff. He was a Russian working in the Pasteur Institute in Paris, and he won the Nobel Prize for his discovery of phagocytosis. And I guess like many great scientists later on in their careers, Metchnikoff would start to come up with some crazy ideas. And he was full of crazy ideas. But one of his crazy ideas is, why did people in what's now parts of rural Bulgaria, why do they live longer and live better? And so he put it down to what they ate. And what they ate was a lot of fermented foods containing lactic acid bacteria. So not only is he the father of immunology, he's also the father of geriatric science and also the father of probiotics. And if you go to Korea, you can get a yogurt drink with his picture on it. So it must have that Nobel sign of approval and must be really good for you overall. And so most of what Metchnikoff talked about has been forgotten for 70 or 80 years, but it's only in the last decades that we began to really appreciate that maybe he was onto something. And that's become with the advent of our understanding of the microbiome in all aspects of health, and including in brain health. And we have two big challenges in the microbiome gut-brain axis field. One is to understand the mechanisms underpinning how microbes could signal to the brain, and that's something we're working on very much. And then secondly, I'm going to share with you some of them marvelously wonderful data we've had in mice, but how much of this can we translate to the human condition? And I also want to remind you that we need to put things within an evolutionary framework and to realize that we're not just our own genes, but we are the combination of our genes and our microbial genes. And indeed, our microbial genes make up 99% of all the genes we have. And this hologenome has to navigate an environmental metagenome, if you want. So understanding this will give us a better idea about microbe-brain interactions across the lifespan. And that's what my lab is really interested in, is trying to understand the mechanisms and the human translation in early life, in adolescence, and in aging. In today's talk, we're going to focus on aging. And, you know, my neuroscience colleagues, well, they're a kind of a skeptical bunch. So they need real hard evidence that the microbiome could be involved in brain health and brain aging. And so how do we get evidence that something is important? Well, we take it out and see what happens. And so the concept of being germ-free, well, that's not new at all. It goes back to Louis Pasteur's time. These germ-free mice allow us to answer the question, is the microbiome involved, yes or no? But they lack any human translation, with the exception being the boy in the bubble that Paul Simon sang about. We use these mice in a bubble a lot, and they've taken us to lots of weird and wonderful places overall. And indeed, the study of germ-free animals in the context of longevity and aging isn't new at all, going back to studies from the 1950s and 1960s, showing that germ-free mice live longer, so without microbes. Now, it reminds me of some of the stories about the nuns who live in enclosed convents who live longer as well. So they may live longer, but they're probably not having as much fun as everyone else overall. And we're beginning to understand, if you grow up without microbes in your gut, what processes in the brain are affected. And one of the most remarkable findings we found some years ago was that the myelination, which is the myelin being the insulation to allow nerve cells to appropriately conduct, that there was a hypermyelination in the prefrontal cortex of the brain, and that this was sex dependent. It was only in males, but it was very, very striking finding, because we mainly think about myelination in the context of demyelinating disorders like multiple sclerosis. But here we have hypermyelination. Adult hippocampal neurogenesis, the birth of new neurons that occurs throughout the entire lifespan. Here we're able to show that in germ-free animals that there's a dysregulation of the brake on this process, and this process underpins higher cognitive function like pattern separation. It's also involved in the stress response. And so again, we're finding clear links between the microbiome and the brain function. We get changes in a variety of neurotransmitters, including GABA and serotonin, that are involved in plasticity in a variety of brain areas, and changes in proteins involved like brain-derived neurotrophic factor, changes in neural arborization. At the neuroimmune level, work from Marco Prentz's group and also from Sonia Garal and others have shown that the brain's immune cells don't function appropriately in the absence of microbes. And work from the Karlinska, from Sam Pedersen and colleagues has shown that the blood-brain barrier integrity is disrupted in germ-free animals. Here's more recent data. We took a slice of the hippocampus being the brain area involved in cognitive processes and learning and memory. And we looked at long-term potentiation, which is a cellular parallel of learning and memory. And what we find is that in male animals, that if you're germ-free, you're not able, here in light blue, you're not able to mount an LTP response to a stimulation. So this is quite remarkable, again, that you need microbes in your gut for such effects. And the fact that there's sex differences, we find time and time again. And for reasons we don't fully appreciate, but it deserves a lot more attention to understand, well, what is it about the male brain in particular that makes it more sensitive to a disruption of signals coming from the microbes? Or what is it about the female brain that makes it more resilient? Now, you don't need to be a neuroscientist to see here. Here is a nerve cell from the amygdala, a region of the brain involved in fear learning, pain processing, social behavior. And you can see that it's already like in a chronic stress state, increased arborization. And this translates to deficits in these types of behaviors. Most notably, we found that in these mice that there's deficits in social behavior. Mice, like humans, are quite social. So if you give them the chance to spend time with a known mouse versus a object, they'll go towards the mouse. Mice, like, well, maybe some humans can be a bit fickle. So if you give them the chance to spend time with their everyday playmate versus a new playmate, they'll gravitate towards a new playmate, but not if they're germ-free. We've also shown that disruption in early life by mode of delivery also affects the social behavior, as does giving antibiotics early in life can lead to long-lasting perturbations. So we're beginning to see very clear links between the microbiome and social behavior. And so this led me some years ago with my postdoc, Roman Stilling, to kind of riff on this. And why is it that we need microbes in the lumen of our gut for proper social function? And you start asking questions about, well, what's in it for the microbes? And you can start to think, if we are in a social, and of course biologists know a lot about this, and everyone does now in the COVID, if we are in a place where there is social no social distancing and we are together, then microbes can get passed on from one to the other. So I, approached a colleague in Vanderbilt, Seth Bordenstein, and I put some of these ideas and I said, like, in terms of evolutionary processes, would there be any value in considering what's in it for the microbes? And he told me that I was completely wrong. And every now and then as a PI, it's good to be told you're wrong. And the reason I'm wrong is that I've forgotten of something that's really important, is that the microbes were there first. And so there never has been a time where the brain has existed without microbial signals. And so we need to really understand that our brains have evolved with these friends, and these friends give us benefits, and in particular, they seem to give us social benefits. So if we explore across the animal kingdom, whether it's in insects all the way up to baboons in the wild, and we disrupt social structures, that we tend to disrupt the composition of the microbiome. And the opposite is true. If we interfere with the microbiome, whether you're a honeybee, a koala, or a hyena, you will affect social behavior. And so we're beginning to really put a framework that there's something about the social brain in particular that is sensitive to microbial signals. And we need to understand what are the factors that might influence that. Ranges from host genetics, sex, social interactions, vertical transmission, the diets we have, the medications we take, and the environments we live in, all can influence this. And then the other part that we're really puzzled about right now is how much is this going to play a role in normal social behavior, in terms of how we trust other people, in terms of how we are acting in a cooperative or competitive way, in terms of the empathy we show and the social decisions that we make. How much is our microbiome playing a role in this? And could the microbiome be the real social network? That is provocative where we are. And in terms of understanding this, we're really trying to appreciate it across the lifespan and understanding the neural underpinnings and the microbial underpinnings of these social circuits overall. And as I mentioned, we're beginning to really appreciate the impact, kind of revisiting a lot of what Metchnikoff was talking about of the microbiome in terms of aging. And almost all systems in the body are affected by aging, and there has been some influence of the microbiome in driving that process. Most notably is work from my colleagues here in Cork, Paul O'Toole and colleagues, which showed that in elderly individuals, the microbiome diversity decreases, and this correlates with health outcomes like frailty. So we wanted to revisit these Metchnikoffian ideas, and we looked at elderly mice, and we were able to show that there's increases in intestinal permeability that correlates with some of the changes in learning and memory, in social behavior and anxiety. We get also an increase in pro-inflammatory cytokines and a change at the microbiome level. So this is quite interesting overall. But we wanted to see, could we do anything about it? And for this, I decided to focus, perhaps for obvious reasons, decided to focus on middle age. And so we took middle-aged mice, and we were able to see already in the brains of middle-aged mice there is an increased neuroinflammation, increased stress-induced immune priming, increased infiltration of inflammatory monocytes, increased microglia activation. And so then we looked at a diet enriched in inulin, which is a prebiotic, and we wanted to see, could the inulin attenuate the effects of middle age, and indeed it could. Now, inulin is present in many vegetables, including leeks, chicory, artichokes, and so really, and this is work that was carried out by a postdoc in the lab, Marcus Boehm, together with my colleague Harriet Shellikins and my clinical colleague Ted Dinan. And really the take-home message here is that if you're thinking of having a midlife crisis, maybe you should forget about the motorbikes and turn to the leeks instead overall. So this was really interesting because it means we can target the microbiome to slow down the effects of aging. But of course, there could be other ways that these prebiotics are having influence that are not just dependent on the microbiome. So we wanted to test this. We wanted to see and do kind of what a killer experiment in aging is to see, is the aging process dependent on the microbiome? So what we did was we looked at the microbiome in elderly mice. These are 22-month-old mice, and we find that it's quite different to young mice. And then therefore, we transplanted the microbes from young animals into the aged animals. And that kind of was a key experiment for us to be able to causally prove whether the microbiome is involved in brain aging or not. And what we found was quite remarkable. We found that there were selective effects, differences of this transplant in animals that received it versus those that didn't. And what we find is that there is changes, of course, in the microbiome composition, changes in the metabolic capacity of these microbes, but at the peripheral immune level, we get changes in a variety of immune parameters, basically making them more like a younger mouse. Again, at the brain level, changes in microglia inflammations. They're not in an activated state, and altered expression within the microglia sensome. At the hippocampus, this brain area involved in learning and memory, we find there are widespread changes in metabolites, and this was also reversed by taking microbes from young animals, and most notably in terms of behavior, we were able to recover some of the deficits in learning and memory and anxiety. Now, many things we looked at, from telomeres to neurogenesis to other aspects of peripheral immunity, were not changed, but it still is remarkable those that did. And really what we're trying to dig into now is, what are the mechanisms? How do microbes signal to the brain? And we have a long way to go on it, but it's something we're very excited about. One way that's quite a strong candidate area is the vagus nerve. And so we showed many years ago, together with John Bienenstock's group in Canada, that when you cut the vagus nerve, all of the effects of a specific bacteria were gone. And this tells us that what happens in vagus doesn't just stay in vagus, but will affect our emotions in some way. We've also, it's worth noting that the microbiome is like a little factory producing all kinds of weird and wonderful chemicals that our bodies wouldn't be able to make. And that's another approach that we've been looking at, and to see, can we just bypass the microbes and go and investigate these chemicals like short chain fatty acids themselves. And indeed we could. In this paper, we were able to show that giving animals short chain fatty acids reversed the effects of chronic stress overall. All of this is leading up to the whole concept of psychobiotics, which is targeting the microbiome for mental health benefits. This is a term we've coined here in Cork, and it's one we're very excited about. There are different approaches, many of them we've already mentioned, from probiotics, prebiotics, fermented foods, but also combinations of pro and prebiotics, as well as postbiotics, which are the metabolites, and paraprobiotics, where maybe they don't even need to be alive. Maybe it's what's decorating their cell wall and what's activating the immune system. We think we're at the beginning of a kind of a psychobiotic revolution, and we've written about this for the lay public to kind of educate people that for appropriate brain health, we need to look after our gut. We've got really good data coming through in animal models looking at specific strains of bacteria and other psychobiotic approaches and how they can affect behavior and also affect the brain. And we're also moving towards human translation. This is a study where we showed that when we took people into the lab and stressed them, we were able to diminish this stress by giving a biflongum 1714. This was reproduced more recently by Paul Inck's group in Tübingen in Germany, where they showed using brain imaging the same ability of a specific strain of bacteria to be able to do this. And this was really gratifying for me as a basic scientist, to go from selecting a strain, testing it in animal models, and going all the way to humans and showing beneficial activity. And so then we took our most active strain of bacteria and to see if it could reproduce these findings, and we cannot get more negative findings into the one paper if we tried. And so that's not specific to the microbiome field or the probiotic field, but it highlights the challenges we have. Nonetheless, when you start to lump things together, and I hate lumping, but sometimes you have to do it, you do see a signal for probiotics and prebiotics in terms of their beneficial effects for mood and anxiety. We're also interested very much in sleep, and we showed that during a chronic stressful period, exam period, we're able to attenuate sleep deficits. And we're also, the whole sleep field is waking up to the concept of the microbiome gut-brain axis, and that's quite exciting. metabolism. And also we're beginning to mine the data that's available there across psychiatry, and you can start to see what metabolites are playing a role, whether it's involved in tryptophan metabolism, short-chain fatty acids, or bile acids. And again, in this paper just out in the last month in JAMA Psychiatry, they did a similar approach, trying to see if there's transdiagnostic aspects and is there a signal of what's going on in the microbiome that might be good for mental health overall. Last thing I want to mention is about diet, and diet can really have a positive effect on this and with the whole area of nutritional psychiatry. And we're trying to triangulate the impact of diet, microbiome, and brain health. And this is work from Kirsten, Birding, and Harold, where we show if we ramp up fiber intake, we're able to decrease stress in healthy individuals. And this is really important to show that our state of gut will markedly affect our state of mind. And likewise, there's large-scale studies, again, this is from Paul O'Toole, showing that Mediterranean diet interventions will affect the microbiome and reduce frailty and improve health status in elderly individuals. And this may have implications for HIV and for all aspects of the healthy brain as we move forward. And it's important to see that the neurology field is waking up to this. Finally, also just to mention that drugs, we're really excited about the impact, understanding the impact that drugs have in the whole field of pharmacomicrobiome and how that feeds back onto the brain. And we think that's really important. It will be important also in HIV management overall. So there's a long way to go in this field, you know, a lot of innovation, but really what we need to understand is the mechanisms at play, understanding more parts of the microbiome than just the bacteria. So the bacteriophage are going to play a role, and we need to understand really how we move the field forward. But the most notable thing is that unlike your genome, which all you can do is blame your parents or your grandparents, your microbiome is potentially modifiable. And that's really good news for your parents, for your patients in general. So here we have a wonderful team of people here in Cork, and I leave you with the words of Hippocrates again, who's taught to have said, let food be thy medicine. Well, I'm going to change that to let food for your microbes be medicine for the mind, and remind you that every now and then it's good to turn things upside down because you'll see them in a different way. Thank you.